I'm Tom Dill. Hello, everybody, especially you new folks. I'm the programs chair for the Dallas Paleo Society. So um, I contact various speakers and get them to come and give talks. And of course, lately we're doing them all via Zoom. So we're getting a few more remote talks. This one is going to be by our local paleontologist and advisor, Dr. Louis Jacobs. Um, uh, uh, Dr. Jacobs, I didn't realize this until I looked at his bio information, but he started out at University of Southwestern Louisiana after a short stint in the offshore seismic work and even the merchant marine, it said. That's where he got his bachelor's degree in 1970, and then he went to University of Arizona. I was aware of that, where he got his master's in 1973 and a PhD in 1977. He then served as a geologist for the U.S. Geological Survey and as a research paleontologist for the Museum of Northern Arizona in Flagstaff. And those of you who remember our last month's speaker, Dr. Jim Kirkland, he also had a Northern Arizona connection, and I didn't realize that Jim told me that Lou Jacobs was his first boss in paleontology, and he thought you were very instrumental in getting him started. He was glad that you were there to help him get started. Anyway, so yeah, weird connections between people. Dr. Jacobs then became the head of the paleontology division of the National Museum of Kenya I assume that was in Nairobi, Kenya in 1981. And since then, all the time I've known him, he seems to have had an interest in the paleontology and anthropology of Africa, including writing a book, Quest for the African Dinosaurs. I don't know if you've seen that, but I recommend that if you haven't had a chance to read it yet. In 1983, he came here to Dallas, moved to Southern Methodist University, where he's been ever since. Started as assistant professor and then just recently retired as full professor. But he's also, and he still is, the president of the Institute for the Study of Earth and Man, kind of a separate research institute there at SMU. All the time I've known him, he's been very active in promoting the Dallas Paleo Society and supporting us. He serves as one of our faculty advisors, try to keep us on the straight and narrow, which is good. And he speaks to our society a lot of times, but he's always usually tries to defer and said, when I ask him, he'd try to defer and say, I have a student that I'd like to give a talk, and I like that as well. We've given scholarships to many of the students, and those students have given us some excellent presentations over the years. Of course, he's done lots of scientific papers, posters, and abstracts, and I didn't even try to count them this time. He did this 2-book, One Question for African Dinosaurs I told you about, and he also wrote Lone Star Dinosaurs, which I think we used to have for sale on our website. I don't know if we still do. Anyway, and then he served as president of the Society of Vertebrate Paleontology from 96 to 98, and then helped bring the annual meeting of the Society of Vertebrate Paleontology to Dallas in 2015. That was their 75th annual meeting. And when I asked him for some topics, he suggested this topic about 9 species of fossils that it turns out they were all named for him. Then just last meeting, I was talking, or a couple of weeks ago, I was talking to people at the Denver WIPS group, the Western Interior Paleontology Society, kind of a sister group of us. And they said, oh yeah, we know Dr. Jacobs. He's writing us a chapter on 9 fossils that have been named after us. So again, it's a real small world in paleontology. So anyway, please welcome Dr. Louis Jacobs for his talk, 9 Eponyms, Stories Behind the Names. And I will now shut up as soon as I hear him come on. Dr. Jacobs,
right.
Are you there?
I am here. I'm here. First, let me say something about Jim Kirkland. Jim Kirkland had just come out to Flagstaff from Boston, and he was a very interesting person, and he was interested in everything, and he's really become a star. So that's great. It's really good you had him talk. Now, with respect to whips, I was contacted by whips, and they said, we want to get everybody associated with whips that has had something named after them to write something up about it. So, you feel a little bit kind of sheepish to be driven down that vain avenue. But I said I'd do it. I don't know whether they like it or not. But then you contacted me, and I think Paulson sicked you on me.
He did.
Yeah, but I didn't want to do what he was talking about. And I thought that if I was going to do this for whips, it wouldn't be right if I didn't do it for you. So that's why I decided on this, and I made it better than the one for whips. And I mean that in a good way for you whips people that are watching, and I see you are. But I had a little bit more time to work on it. So what I'm going to do, I'm going to try and share screen. So I'm going to press that button. The title of this talk is on the next slide, but I wanted to be able to define what an eponym was. So an eponym is a person, a place, or a thing after whom or which someone or something is or is believed to be named. And, I think that would fit all of us.
Sure.
So 9 eponyms, stories behind the names. And these are, I don't know what was going through the minds of the people that named these things. But I've got them listed on this title slide. They're just in alphabetical order there, 9 of them. And you see most of them are rodents. And I don't know whether that's their statement about my character or what, but that's what we've got there. It's a pretty good spread in terms of age distribution and geographic distribution. And I'm really going to tell you about the science behind what was going on when these things got named, because I thought that would be more informative, and I know it would be more fun for me. So I'm going to start out with my master's thesis, which I published in 1977. Okay, so this is the San Pedro Valley of Arizona, and my thesis was on the rodents of the Hempylian age, that's Pliocene age, Redington local fauna of the San Pedro Valley, Arizona. And this was a class field trip where I went in late 1970 or early 1971. And this is what I did my thesis on. I'm going to tell you more about it, but Charles Frick used to be a very rich fossil person, and I'm going to tell you more about him in just a second. But in this master's thesis, I screened this quarry, and I had six new taxa. 3 new genera and 6 new species, which is not bad for a master's thesis. And I've got them listed on the right-hand corner, that Ronchiomys and Galuciomes, those that are bold-faced. Those have been used to characterize larger groups, not just species. So, now there are Ronchiomines and Galuciomines and stuff like that. So that's pretty good. But there's a very good paleontologist by the name of Tom Kelly, and he took one of the things I had named, Paronychomys, and named it Paronychomys Jacob's Eye in 2013, and it's from Smith Valley, Nevada. So Whips wanted pictures of the field and where it came from and all that. So I tried to get Tom to send me a picture, and he didn't have any pictures. And and he didn't have any pictures of the site. So I chased down a picture of Smith Valley from the Los Angeles County Museum, and that's Smith Valley, and that's the site. But the scale is so large you can't tell. What you can tell is the picture that Tom sent me that is the side view of a complete skull. And I don't think while I've been screening, I have ever collected a good fossil like that. At any rate, this brings up the questions, why should you study small mammals? And the reason is they're not as well known, and so therefore you can find new ones. And there's a lot of different species of them. There's a lot of different species of them right now. So for instance, if you asked how many living species of horses are there, you would say 7. And if you said how many living species of mice are there, you'd say more than 1,500. And since horses and mice evolved about the same time, you can tell which one evolved faster, and therefore they help you tell time geologically. And that's going to underlie this quite a bit.
It smells really good.
Where is this? Oh. Come on. All right. So this is the picture I showed you in the lower left is the picture I showed you before, and that's the class field trip. If you go to the top left, that is pulled out a long ways, but it's the same site. And if you go over along the cliff face there, that's where we were excavating. But you see all this big mound all out here? That is all a spoil pile from when the Childs Frick's collectors were collecting fossils from this site. And I mean collecting fossils. They were getting camels and horses and big carnivores and all kinds of things. And it was worked by a fellow by the name of Ted Galusha, who was a collector for Frick. Frick had teams collecting year-round. They'd collect in the warmer latitudes during the winter and in cooler latitudes during the summer. And Ted Galusha told me that he had moved that cliff face back by about 20 or 30 meters. So that is an absolutely incredible effort. They used dynamite. It was industrial grade mining for fossils. And that is what Childs Frick wanted to do. And he ran several crews. So the Frick collection of fossil mammals, when he died, went to the American Museum of Natural History. And it numbers about 200, 300,000 specimens and takes up 10 floors at the American Museum of Natural History. But Frick was very secretive, and they would backfill their quarries at the end of each season. Have any of you ever heard of this? I mean, I tried to get pictures of Ted Galusha in the field and get some actual shots of them digging and excavating from the archives at the American Museum, but I didn't have any success in it at all. But at any rate, I do have a picture of Ted Galusha and his wife, Marion, and were both very nice people. And Ted's the one that told me he had moved that face back by 20 or 30 meters. It's just an amazing chapter of paleontology that you just don't know anything about because Frick was so secretive. One of the advisors in my graduate work was George Gaylord Simpson, who had been at the American Museum. And Frick didn't like him and wouldn't let him see any fossils. So some of those collectors like Ted Galusha would sneak him in where he could get a look from time to time. Now, I named a rat after Ted Galusha, and I have a picture of a broken tooth down there. It's not a very good picture, but I did it myself, and that's what I could do. The lower right-hand part, that's, these teeth sit on the head of a pin. They pick them out isolated, mount them onto a head of a pin that's stuck into a cork, and then you can stick the cork into a glass vial, and it's supposed to protect it. So I got this new fossil, and they actually had a couple of upper teeth in the Frick collection at the American Museum. As a matter of fact, two of the things I named have holotypes in the Frick collection. But the reason that this tooth is broken is because when I had finished my thesis and I was sitting down with my professor, Everett Lindsay, for him to look at the fossils and look at what I had said, he was sitting there and he was very, he was just stone-faced, and he just didn't talk much. And I was a nervous graduate student, and I was just sitting there next to him, and there was no talking, and it was quiet. And he looked at it and looked at it and looked at it, and then he handed it back to me and he said, "I think you're right." And I was so excited, I took the cork in my hand and I was putting it back into the vial, and I smashed it into the bottom of the vial, and that's why it's broken. So my professor got a dustpan and a whisk broom. He got down on his knees on the floor and he swept it all up, and he took it back into his office and looked under the microscope, and then he put all those pieces together, and he came back out and he said, "I couldn't find that one corner." So there it is. Isn't that interesting? Okay, now here's the thing. Very many problems in earth sciences rest on resolving geologic time. And that professor who fixed that broken specimen was Everett Lindsay. And he was very interested in geologic time and using fossil vertebrates. And he was working with a couple of geophysicists. And at that time, everyone knew that the Earth's magnetic field changed in the direction it pointed, and they were trying to figure out how you could use those changes to tell time. So they were starting in the San Pedro Valley of Arizona, and that's why they directed me to work, or that's why I was encouraged to work on that site. So this is their first paper, which was really a landmark paper. And throughout this, I hope you don't get bored, but it's very important to show these logs where they're black. That means it's normal magnetic direction, like where the North Pole is now, your compass would point, and where it's white, it's reversed. So it's the pattern that matters. And if you can date any of these flip-flops, then you can correlate it all over the world, and that's why it's so important. So that brings us to Mexico. That's me down in that wash. This is at Yapoma, Mexico. And that tooth that's inset there, that tooth is a genus that is called Jacob's Omes, that was named by a friend, Nick Chapluski. When I finished he's at University of Arizona, Oklahoma now. So when I finished it at Arizona, I went up to Flagstaff to the Museum of Northern Arizona, but we were still working down in Mexico. And Nick, I had gotten a call that fossils were found in the Verde Valley. So I went down there and checked him out, and Nick was a student at the University of Northern Arizona, and he took that on and he did a great project. And in the upper right, that's Kyle Davies from University of Oklahoma at one of the quarries in the Verde Valley. Now, Nick was very gracious in naming Jacob's Omies for me. And he told me that I was helpful to him when he was a student. I don't remember that so much, but I'm going to take his word for it. But one of the things that Nick did is, you see where it's quarried out underneath Kyle's arm there. And if you look down in the lower right, he took out big slabs of this um, mudstone and prepared the surfaces, and what you see corresponds very well to what you would find underneath a modern owl roost. So he got just a terrific set of fossils that he's done a lot with, and it's just great taphonomy. Everything's really good what he did with it. So now, from starting in the San Pedro Valley, we've been to Mexico and the Verde Valley. So why were we doing that? And it goes back to that same reason, that PaleoMag, because those same three guys, including my professor, they were walking right across the country, going to all the appropriate places where you could do the PaleoMag of that age. So there's the Verde Valley, where Jacob's Omies from. This place in Chihuahua, that's Mexico, and we named some stuff from there having to do with the origin of South American radiation of rodents. That's the San Pedro Valley. Oops, sorry. That's in northwestern Arizona. That's in up by Santa Fe, and then Hemphill, Texas, and White Cone is also in Arizona. So using that PaleoMag and the stage of evolution of all those little mammals, then these correlations could be made, and that was a big deal in those days. So we tried to do the same thing around the Cretaceous Tertiary boundary and on up in the San Juan Basin. So in the upper left, this is our crew in 1975. That's me with the John Deere hat. And then Lou Taylor, who's a big gun in whips. He has had a lot to do with that for many years. Jim Honey, who also used to live in Denver. And then Larry Flynn, who's at Harvard now. So the reason we're sitting there, we're sitting in a quarry where we had just dug up a skull of what was thought at that time to be a horse ancestor called Tetraclinodon. And Jim Honey, you can see on his finger, he's got that bandage. And that's because he whacked himself in the finger when he was trying to dig it out. And none of us were looking, but he grabbed his hand and pulled it up to his face, which splashed blood all over his face. And when we got to him, we thought he'd hit himself with his own hammer, but he recovered nicely. Anyway, that's in the Middle Paleocene, Typtorium there. But we went all the way up section, and on the right, you're seeing outcrops of the San Jose Formation, which is Middle Eocene. And there's an image of a tooth there. That's the upper first molar of something that was named Apatosaurus jacobsi by Kathy Flanagan, who was one of our colleagues there. And then just to prove we hadn't forgotten this PaleoMag, that's the logs there on the left. Now, this one's a good story because there was this fella, Li Chuangqi from China. I met him when I was a graduate student because he came over to see my professor and to see Dr. Simpson. And they were busy, and I was given the task of showing him around. And he just the most wonderful guy. He was much more senior than we were. So the graduate students have for decades called him Uncle Lee, but it came from that. And by the way, that picture in the upper right, that's at Chukutien, which is where the caves are, the karst where Peking man comes from. So Uncle Lee and I named a new species from the Yusha Basin, and we named it Chardonnomys yushiensis. Now, we named, and there's the picture in the lower left inset. That's the tooth of that particular species. But it turns out if you go to the Yusha Basin and you collect layer by layer, there's lots of Chardonnomys. One of those had been named Chardonnomys lewisi, but our good friends Larry Flynn and Will Downs went over there and started collecting layer by layer. So my good friends proved that Chardonnomys lewisi was not a good species at all. One of the interesting things about that is that the reason they were there anyway is because that Childs Frick, that industrial grade fossil collector, had made huge collections that are now in the American Museum from Yusha, and Larry and Will had gone over there to follow that up. So now we're going to go to Pakistan. And if you look in the upper left, the green country is Pakistan. India is the orange country. The subcontinent of India, which is what those two things are, that's, whoops, you were talking about that earlier. That drifted off in the Jurassic and came up north, and in the very early Tertiary slammed into Asia, and the Tibetan Plateau and Himalaya were raised up. So that's still going. If you lay in your sleeping bag at night and it's quiet, you can feel yourself being raised up by the earth growing. That's not really true, but I can't see your eyes, so I don't know whether you think it's amusing or not. At any rate, you see in yellow there the country of Pakistan, and then that red is the Potwar Plateau. So the Potwar Plateau exposes a lot of rock that is named the Sewalik group, and there are lots of fossils, and it's thousands of meters thick, and it ranges in age from about 18 million to 2 million years old. So since the early 70s, there's been a big project headquartered at Harvard that has been working on those Sewalik rocks, and it's still going on. So it's done amazing things, and I'm gonna walk you through what I did with that stuff. And I think that's interesting because it tells us a bit about how the science has changed, or at least how I've seen it changed. In the lower right is the skull of Sevapithecus that the project found. And you know, the study of human evolution is always a driver of research projects where they occur. So when this first started, Sevapithecus and Ramapithecus were thought to be more closely related to human evolution than is thought now. And they're more thought to be like more related to orangutans. So this is Bonnie and I and the Sewaliks. And you see that those beds are dipping. They're dipping because India slammed into Asia. And you see the dip slope we're standing on, and then you see the valley, and then you see the dip slope in the far distance, and then there's more of those all the way down. So that's Sewalik group rocks, and they're just they're just an amazing geological wonder. Now here you see there is somebody collecting sediments for screening for small mammals. And here is somebody collecting sediments for screening for small mammals. So that's the way it's done, just stepping through the entire stratigraphic section, collecting all along the way. Now, my responsibility was those small mammals, which in the Neogene is mainly rodents, but people were doing and still are doing everything. And our first stab at things in 1984, you can see on the left-hand side, you can see our magnetic log, but we also have the ranges of all kinds of animals in there. And that's been improved and perfected since 1984 when that was published. But isn't that amazing? Now the difference in time, because of the PaleoMag and being able to work out sedimentation rates and the like, then for that amount of time that we have, especially from about 16 to 8 or 6 million years, you can break it all down into 100,000 year packets, which is amazingly precise for the fossil record. And the amount of time between these two localities, the lower one and the one above that sandstone there, is about 80,000 years. So that's pretty good. Now, that brings us to Carnimata jacobsi. I did my dissertation on stuff I was finding in Pakistan from 4 localities. And the screening was done. I'll show you how I originally did it, but Will Downs took over most of the responsibility for most of the years after I didn't go as much. So you can see he's in the oops, I'm sorry. Got a hair trigger on this thing. Okay, so Will is standing there in his Pakistani garb, and there's a camel and the donkeys that had brought sediment. You can see sediment drying on the plastic, and you see these tandem screen boxes. And here at this camp, and because there was water available, then it was being washed in buckets. His assistants, because they washed the dirt in buckets, became the buckettes. And that is Michelle Morgan. And in the middle is Elisa Winkler, who you all know and she's talked to you. And she's very accomplished person at Southwest Medical School. And then Catherine Badgley of the University of Michigan following her. So they enjoyed their work, I think. And Elisa did her dissertation with me, only she was working in Africa. So she named Carnimata jacobsi from Kenya, but I named the genus Carnimata, and I named it after this picture on the right that I saw in National Geographic in 1977. And that is a picture of the temple of Carnimata, which is the god of rats. And you see all these rats being fed down here. And there's even one peeking over there wanting to go in. So I saw this when I was trying to think of what to name these new things. So here are first upper molars of 2 species of Carnimata. On the left is Carnimata darwini, which is a little older than Carnimata huxleyi. And then Elisa found it in Africa, which is a good biogeographic story. And it's so good, it's better when you include with it, if you include with it the fact that elephants went from Africa to Asia earlier than Carnimata went to Africa. So on the left, this is Ganesha, the elephant-headed god. And on all the Ganesha statues, you see there's a mouse down there at the bottom. And that's because everyone knows that mice can go anywhere. They can even go into walls. So for Ganesh to get into everywhere Ganesha needs to go to do the godly things, always rides on a mouse. So with the oldest known elephants, proboscideans from the Indian subcontinent, we got fossil rodents. And so we named the fossil rodents after that mouse. So we named it Baluchimese because it's from Baluchistan, and Ganesha fur because that means Ganesha bearer. So while Ganesh came one way, a little bit later, Karni Mata, as we know from Elise's work, went the other direction. Now, when I first went over there, the first few years, I stayed in a village called Padre, and everybody else would come back, and I'd stay there a few weeks longer to screen wash. And I was washing in a little pond not far from the village of Padre. So here you see the process. These are my colleagues. This on top, getting sediment from that locality. That's Saeed Alizai, who went on to become a geologist in Scotland. And then we had a camel walla that would help us and load the camels with the sediment we wanted, and then bring it back to the washing pond and unload it there, and then we would be able to do the screening. So you don't see any water flowing into that pond, but I can tell you it stayed pretty, the level stayed pretty constant. And I always wondered why that was, but every day at a certain time, a flock of goats would come and have a drink and then pee in it again, and it would always stay the same level. Well, that was okay, except if you go to the bottom left hand, that fellow there, Abdul, he would come down from the village and build a little fire, and we'd have lunch together. And we had a watchman stay in that tent, so just to watch over the equipment. So one day after he heated up the meal, he said, "Would you like to have a cup of tea?" And I said, "Yeah, that sounds good." So he walked over and got the water out of this pond, and I decided I really didn't want tea that bad anyway. But we got amazing things from this locality. And what you see on the inset in the lower left of the right-hand picture are two teeth. One is the oldest species of the genus Mus, which is house mouse or laboratory white mouse. And then on the right that's 6 million, and then there's a 2 million on the right. So what we're doing there, if we're right about that stuff, is that we're bringing the fossil record right up to species that are alive now. And they're not just any species, they belong to the most abundant group of mammals living in the world today. And there's more known about them than any other mammals except humans, because they're used as laboratory animals. So after that, there was a fellow named B.S. Kotlia from India that named Mus Jacob's eye after me. And his diagram is on the right. And the first two things down there are what I had named, and then he's got Mus Jacob's eye in there. It was a nice thing for him to do. Then we went younger to about 2 million years old. And I thought we had something that was like Rattus, which is the other white rodent that's used in a lab. And that was an important thing because they were both laboratory animals, so there were a lot of experiments going on with them at the time. But at any rate, in the upper left-hand corner there, that is, those sediments are in the Pobby Hills. And the Pobby Hills, Pobby means arch of the foot, so it's an anticline, basically. And if you look far in the distance, you can see the snow-capped Himalaya there. But that always struck me as being like a needle in a haystack, because it was such a vast area. And for this particular animal, I only had 3 teeth, and there were 3 or 4 other different kinds, but none of them with more than that many teeth. So I always liked the picture in the lower left. Those were our 2 drivers. And I can just imagine them thinking this is like looking for a needle in a haystack, which it pretty much was. So I said this tooth that you see inset up there was related to Rattus. But Guy Musser of the American Museum of Natural History said, no, that is Hadramese Lou-Jacobsi. And he was right. I mean, he had a very good paper, and I was wrong, and but it was a really good contribution he made. This picture, Hadramese is still living today, and the reconstruction there is the 19th century reconstruction of the living animal. So now let me put this all together for you.
Before you go much farther, Dr. Jacobs, I noticed you were coughing there a little bit. Would you like a tea?
I'm good. I'm good. I've only got another 3 hours.
Oh, okay. Well, I thought if you, I was going to joke I didn't make it out of the water hole, but if you want to get yourself a water bottle or a drink of water, I thought I'd give you a chance to take a break if you felt like you needed it. You okay?
Well, thank you very much.
Okay.
All right. So my dissertation on the upper left, my dissertation was, that's the cover of it, Fossil Rodents, 2 families, Rhizomyidae and Muridae from Neogene zoologic deposits of Pakistan. Now, just go straight on over to the right there, and you see a diagram that I would never let my students draw. But that's the way I tried to figure it out in those days. And the things that I can tell you about it is that I had 4 stratigraphically superposed localities, only 4. And from that, I worked out what I thought the evolutionary relationships were. And in the paper, I have a table of characters, and I have the numbers on there that says where I think they go. So, you know, that's how I did it. Now, let's drop down, and you see something that looks like, I don't know what, but it's got those dotted lines, and there's a list of genera all around the top. Those are living things. And I was trying to express there that from the oldest members of that family that we had in the zoologics, there were 2 major groups that came off. And one of them I thought was Rattus group, because that's what I thought those ones from the Pobby Hills were. And the other I thought was Mus. So that's what I said, and I said it was oversimplified. Now, David Pilbeam, who's the overall leader of this and who's a gigantic human evolution guy, he knew that what was going on at that time was people were using white rats and white mice from the laboratory to look at their molecules, actually their immunological differences, their antigens and antibodies, and try and figure out how old were they, their fossil record would be till their divergence. So what I have in those 2 crummy diagrams on the right made him say, you've got the best evidence there is right now. So from that, we said rats and mice didn't split apart 30 million years ago. It was between 14 and 8 million years ago. Now that was a long time ago. So now, let me just walk you through this real quick. If you look on the lower left, those horizontal colored bars, on the right side of that, you see all those numbers along that axis that says age in millions of years. And what that's really telling you is that instead of the 4 localities that I had when I first wrote that, there's now 45 or so in only 2/3 the time. So it's a very densely sampled. Those horizontal lines that are going from place to place, that's a progression in time of the different identifiable groups that we have and their relative percentages, which at what locality has more or less percentage. And what we're finding is that you can't tell the species apart because they just start blending together. And we know that because if you go up to the top row of charts, graphs, what that's showing you is that you can take individual teeth and you can measure specific characters, and we have enough of a sample at enough levels to actually see how and when the characters diverge for each character. So that's a very different way to approach things than how I did it in 1978. And the significance of this is that increased density of stratigraphic sampling of statistically significant numbers of specimens underpins fossil-based hypotheses of evolution. That's a big statement, you know, because paleontologists went through a number of years where you just weren't allowed to recognize ancestors and descendants. Now, here's another thing. If you get a preferred tree, if you say, I think these are how they're related, and these heavy black bars are what their age ranges are, then you can actually model what the times of their divergence should be, even if you don't know what it is. So the significance of that is that algorithms or equations or statistics can generate hypotheses to stratigraphically test splitting times in a preferred tree, right? And that's how I did it before in the lower right there. So then, if you put it all together and don't try and look at it, but if you look on the right there, you've got my hand that had the 2 major groups. Well, there's still mainly 2 major groups, but this that you can't read, that big column, that is most of all the genera of living members of this family, and their DNA has been done and compared. And the DNA is calibrated by the fossils from the Sewellics, and then you get a molecular prediction of the age, and you can compare it back with the predictions you get from just using the stratigraphic succession. So the significance of this is that the chronological precision and dense sampling in Pakistan results in phylogenetic synergy between molecular biology and paleontology. In this case, between the most diverse group of mammals living or extinct. And if you don't think that has any implications, Google on Google Scholar, Google of mice and men, and you will get so many papers about what the relationship, what mice tell you about people and people tell you about mice, and how they're used for disease treatment and how they're used for biogeography. And all of these things, all of these studies use these divergence times in this record that's been developed. So it's changed a lot. So enough about mice. Now we're going to go back in time to 1977 again. In the upper right, that is the Placerius Quarry, which is a Triassic site in Arizona. And you see quarrying there. I guess I'm taking the picture. That's Larry Flynn in the back with his hat on. We've all been friends forever. That's Bonnie right there digging. And I think Larry's wife is right there also. So again, we weren't doing any paleo mag there, but we were screening. And one of the things we screened and found, if you looked on the lower left, is Trilophosaurus jacobsi. And that Phil Murray, who's an SMU alum, named that, and that's the diagrams from his paper. Then from Texas, the Triassic of Texas, this skull in the black silhouette, the skull was found. And that skull is now on display in the Albuquerque, New Mexico Museum of Natural History in Albuquerque. And they had a guidebook to the display, and that's the reconstruction of Trilophosaurus jacobsi up there. Now, I think that's great that that we found that. That was a new thing. But then there were people more recently were going through the collections, and they found something else in a similar group to Trilophosaurus. It's called Pelacrodon, and sorry. And in the lower right, that's Pelacrodon. So you can see the teeth are kind of similar, but what's interesting about that is it wasn't recognized until 2018. So, you know, the work we did there was in 1977. Well, I guess Phil named it in 1987. So these collections keep paying off. The other interesting thing about Pelacrodon is that it was a genus that was known, but it was known previously from South Africa and Antarctica. So this is a Pangean specimen that is now in the Placerius Quarry of North America and then those two southern continents. Now, I'm showing you pictures, upper left. That's Roy Chapman Andrews' photograph of flaming cliffs in Mongolia. This next fossil is not from Mongolia, but I haven't been where it's from, so I thought I would do Mongolia. So the picture comes out of the new conquest of Central Asia, but I see I put that in backwards. But anyway, that's my copy of New Conquest of Central Asia. And if you look just to the right of that, it was signed on the inside of the cover to Louis Jacobs, the new dinosaur hunter from George Andrews. George Andrews was Roy Chapman Andrews' son, and he used to live here in Dallas. And when Bonnie and I met him and his wife, then he signed that for me. So in later years, I'm standing in front of flaming cliffs with Lu Jun Chang, Yoshi Kobayashi, and Young Nam Lee. These were all three students of mine at SMU. You gave money to all three of them, and I think they all three gave talks. Young Nam Lee named the dinosaur Papasaurus, and he also recognized the pterosaur Calabarhynchus from the Motorola site in Fort Worth. And he did the footprints and named one after Bill Lowe out at Grapevine. Yoshi was studying, he was studying dinosaurs, and so was Jun Chang. Jun Chang was the oviraptor expert, and I forgot the name of what Yoshi was. But they were all very good students, and they met at SMU, and they became fast friends. Jun Chang was Chinese. Yoshi was is Japanese, and uh, Youngnam Lee is Korean. Youngnam Lee is now a professor at Seoul National University, the first vertebrate paleontologist they've ever had. Yoshi is at, um, what's that northern island up there? Hokkaido. Hokkaido National University in Japan, very well respected. Just got a big science award over there. And Jun Chang, a very, very nice fellow. And he was recognized as one of the top 10 researchers in the Chinese Academy of Geological Sciences. So they were all very, very accomplished and were right there at Flaming Cliffs. So they named a dinosaur after me, and they've all got their fingers in it. And this is what one of those children's weeklies said: "Meet Corythoraptor, newly found dinosaur, resembles ostrich gone bad." But what it is really, you know, just remember, I've been showing you isolated rat teeth for all this time. And look how complete this dinosaur specimen is. So this is in the upper left, you see the whole specimen. You see the bones drawn out on it next to it. Go down to the bottom, you get a look at the head and a reconstruction next to that. But if you go to the right, what they're doing, the first row up there, that's the head, and then a diagram of it to make it clearer, and then a reconstruction. So if you go to the second row there, they're looking at the structure of the bone in the top of the head and saying, well, that looks a lot like the bone that you see in these cassowaries. And same with the bottom row. So that's why they reconstruct it like they do. And here you can see the outline of the skull in black. It has its leg thrown up over its head, but you can see there are claws in there, and that's the upper bill, and then the lower jaws here. So there you see the difference, and there's the modern cassowary there. And this is the way they reconstruct the entire dinosaur. Corythosaurus jacobsi. So that's it. Thank you very much.
Well, thank you very much, Dr. Jacobs. Everybody, please give a hand if you can, just on your screen. Thank you so much. Let's take some questions now.
Well, Linda asked a question for Dr. Jacobs, and she is interested in knowing what years you were in Nairobi. You started in 81, but how long were you there?
I think didn't we go in 80? Yeah. We were there, we went in 80, and we were there about 2.5 years, I think.
Excellent.
We moved to Dallas in 83, Bunny said.
While you were there in East Africa, did you ever meet the famous Dr. Louis Leakey?
He had already passed on, but we worked for his son. We worked for Richard Leakey.
Oh, okay. Now, I'm looking for questions, but I've got one in the meantime. Rats and mice. I guess you like to study them because they're so abundant. I got that. And they have lots of small teeth that you can find. But why did you have to go to such exotic places to get a nice, complete record of rats and mice? Could you do that in Montana or Nevada?
Well, here's what was going on at the time. You know, I started out with my thesis in the San Pedro Valley and showed you how those guys were trying to piece things together. In those days, the fact that you could do it in marine sediments had just been figured out, and nobody knew if you could do it in terrestrial sediments. And the reason that it started in the younger rocks is because uh, the development of the time scale started at the younger end of things, because all you had to do was go to a lava, date it, and then you could stack them up. So, and that was all at the young end. So then, once you had the pattern that was definite from the lava, then you match that in the sediments and then work down from there because you had it anchored. Okay, so when we went to the San Juan Basin, we had an anchor which was the Cretaceous Tertiary boundary, and we could work up from that. But you can't go anywhere else in the world, or not many places else in the world, where you have so many millions of years essentially continuously deposited, because when India slammed into Asia and the Himalaya went up and it started eroding down and covering all those carcasses, now they get pushed up and eroding out. So you just have a tremendous thickness. And in those days, the longer section you have, the assumption was the more likely you would be to find more reversals in the Earth's magnetic field. And because the reversals are randomly distributed, then you would be more likely to find a characteristic pattern that you could be sure of. So that's why we went to Pakistan.
Great, thank you. Yeah, so to get a good record, you go next to a giant rising mountain range, I guess, because then that gives you a huge, thick section to work on.
It works there.
Yeah.
And, on the other side of the Alps, you have all of the Süßwasser Molasse in southern Germany and all that area. So,
Great.
source and sink.
Okay. Had a question. Well, I don't know if it's a question or a statement. Fernando, he asked, the Texas Coloborhynchus, was it reassigned to its own genus Uctenodactylus? And Fernando's there. I see his picture right below yours. You can say hi to each other.
Hey,
There was and I think it was one of Kellner's students. I think did that, but I think it switched back again.
Oh,
really?
There's a lot going on in pterosaurs. So
Check that out.
all your eponyms species are from other places. Any interesting Lone Star rodents. First, let me say, I have no control over the eponyms. That is, everybody does their own. But yes, there's lots of interesting things here. As a matter of fact, on one of the slides I showed you when I said we're just going through all these different places and doing the paleomag and matching them up, one of those places was here in Texas. And, there are the type localities of 3 land mammal ages are here in Texas, in the Panhandle. So there's lots of stuff here. But, I still work on the stuff from Pakistan because we've got it, and it's such a fantastically good collection. So,
yeah. Are you you might be referring to the question from Donna Cole? Are there eponyms? How do you say that? Eponymous species? They're all from other places. Are there any interesting Lone Star rodents?
there are lots. Yeah.
Yeah. So we do get some of our own, huh?
Mm-hmm.
Okay. Somebody asked a question, maybe facetiously, but maybe there's something to it that we should address. Should we be marking our finds with the north arrow? I mean, is there a possibility that people could do paleomag on individual fossil specimens?
Well, the first thing is if you're doing a jacket or something like that, you should mark the orientation. And you know, the picture I showed you of Nick Chaplewski's uh, little fossil bones in the Verde Valley.
Mhm.
No, all the orientation, that all goes into the taphonomic interpretation. Uh, as far as the paleomag goes, you would probably want more than just one sample, and you'd probably want it from the sediments around the specimen, because what you're really doing is getting the small iron minerals that orient themselves with the Earth's magnetic field when they fall out of suspension.
Yeah. Okay, thank you. Let's see. Somebody asked, where was that paper on Pakistan published? I think there was the one called on your work in the Sawaliks.
Well, there's a lot of them. If you just send me an email, I can send you PDFs.
Oh, okay. Very much. So it'd be okay if we pass on your email address. Well, they can figure it out if they go to the university, but whoever asked that question, feel free to contact him. He could get you a copy of that paper. Somebody asked, have you assembled any of the mice or rats? Are these things often mounted as specimens? Do they put them back together?
The bones,
Yeah.
we didn't get many bones of them. We mainly got the teeth. Uh, modern ones, you know, modern ones are disease vectors. So, you know, the Department of Defense, that's why the Smithsonian has such a big collection of stuffed mice from all over the world, because they have to know what diseases are there. So they go trap mice and dust all their fleas off and save those and try and figure out what diseases are where. But the fossils themselves, there are some fossil rodents that have been mounted. You know, we don't have any here that I know of. But you have to have a good enough specimen.
Mm-hmm. Yeah. And have there been localities where you could document that it was like an owl pellet? You know, something
That's what Nix does. He has a
Yeah.
paper. He has a paper doing just that.
Okay.
You know, I mean, he shows the fossil concentration, and he shows modern owl pellets.
Certainly not hard to see why in dealing with all those bits and pieces and tiny rodent orts, that once you discovered your dinosaur, mostly complete, not difficult to see the leap to dinosaurs.
Well, when the dinosaurs came about more or less by accident. The Malawisaurus and stuff like that I was looking for Cretaceous mammals, which I got some in Cameroon, but not much, none in Malawi. And here, Proctor, man, that just a gold mine.
And that wasn't then when that described by you guys and Phil Murray. You talked about being your student and
Well, see, it was one of Phil Murray's that found that site out at Lake Proctor.
Yeah. I can't remember what DPS member was talking to me. Somebody was telling me about being out at that lake, and that the citizens of the local town used to just go and sit on those sediments and watch fireworks shows. And then
Well,
they looked around and there were these bones.
Do more than that.
What's that?
They'd go out there and sit and do more than.
Yeah. but is it, I don't know, the story I heard, it was true that they just thought there were chicken bones lying around next to them, and until somebody looked at it a little closer and realized those weren't chickens.
Well, Phil Murray had taken, as Rusty Branch is the student's name. He's a geologist now. but he had gone out in the field with Phil, and so he knew it. He knew what the bones looked like, and he recognized them when he went out there.
Very cool.
Well,
Anybody
nice
else got?
talking to everybody.
Yeah, it's been great having you here, and I'm sorry we can't do it in person. Hopefully, what, 5 or 6 months, whatever. Hopefully, we'll be able to do face-to-face meetings at some time. All right. Well, yes, I want to say, everybody give Lou Jacobs a round of applause around on your screen. Or they used to have a
Thank you,
little
Louis.
applause icon, but they don't have
Hey,
that
Lou.
anymore.
Thank you.
Thank you, Louis.
Thanks.
That was a lot of
Hey.
fun. Yeah, a
Nice
lot
to
of
meet
fun.
you, Miss Taylor.
Nice to meet you.
Question,
Yes.
does Lee Taylor have anything to add to the field stories?
You don't have nearly enough time. I do have a question though. The chart that you just submitted, you and Yuri and Larry, how much time is there in that stratigraphy that you're presenting?
Well, Larry's mainly interested in the 12 to 8.
Okay.
And, the first thing that David and I said, it would be between
Nice.
14 and 8 or whatever we said.
Yeah.
But now it's pretty much narrowed down to 11. So that's what we've been spending a lot of time on.
Do you
And
see?
also,
go ahead.
the small mammal localities aren't evenly spaced. There's some gaps between the PABI and the rest of it.
Do you see in that chart? Do you see evidence of competitive exclusion causing extinction? Or have you thought about that?
Well, Yuri looked at it using laser ablation stable isotope stuff, competitive. Looked at tooth form and carbon isotopes to see if the diets were driving them apart or something.
Okay.
And there's some of that, but it's hard to tell about. Where was I just reading about that?
It came to my mind, I heard Eric Scott give a talk the other day in Blaine Schubert's programs, and he commented that 1,000,000 years seemed like not very much time to drive another, or it seemed like a long time to drive another species to extinction. And I thought about multituberculates and rodents, it was 20 years, 20 million.
You know what? And that's probably what I was revisiting, because Larry cited that in the paper they've got on meta communities or something. And by the way, everybody, Lou Taylor out there in that San Juan Basin, he published on the Paleo Mag out there.
Yeah, we did the Paleo Mag. I collected the samples for your Paleo Mag in Pakistan.
Where are you at, Lou? Just asking.
I'm in Denver.
Oh, okay. And so
Yeah,
now
I'm
you're
affiliated
associated
with the museum in Denver and with Dinosaur Ridge. Been a WIPS
Great.
member since I moved to Denver, I guess, early 80s.
Great. Another contact with the Denver group. That's great.
Mm-hmm.
So I know that this is not,
Larry!
I know that this isn't about paleontology, but they might enjoy the story about the mice on the stick in Malawi.
Well,
The ones you collected for the Department of Antiquities.
well, driving down the street and driving down the highway in Malawi, these kids would catch mice in the field, and they'd boil them and dry them out. They'd leave their heads and their tails and their feet on, and they'd put half a dozen of them or so in split piece of bamboo and you could drive down the street and get your lunch.
Mhm.
So it was when Elisa was doing her work. And so I bought a bunch, and then when I got back to Lilongwe, the people that worked there thought that they had been brought a treat and ate them. I got some more. and got them back. I was on sabbatical at Harvard, so we were living there in Cambridge, and I brought some back, and they started smelling bad. I put them in a plastic bag and tried to hide them in the stairwell without much luck. So I figured if I dried them out, they'd be okay. So I went to Pilbeam's lab and put them in his microwave.
Oh.
A related question about rats and mice. You know, I've heard that wherever humans are, there are rats and mice. You know, they were just they so rapidly adapt to us. Is that true of ancient man sites? You know, like the Leakeys, they're up in the Rift Valleys in Africa. Were there rats and mice living with the humans?
Almost certainly. Those commensals, you know, most that family Muridae, most of those are in Asia and Africa and Australia, essentially all of them. But wherever humans go, you find mus musculus, house mouse, and you find roof rats and Norway rats, brown rats. So they go along with the humans everywhere. And there's evidence, I just saw a paper on that not long ago too. Apparently that kind of commensalism has originated several times. Now, you know, when these people do studies on commensalism, then they do the molecular sequences of the animals they trap, right? And then they ask, how long have these things been separate? And to do that, then they go back to the chronology of the molecular evolution that relies on the fossil dates. So it all ties back to those fossils.
Really cool. Yeah.
Well, thanks for having me. It's good to see you.
Thank you.